Plant tissue culture. Plant tissue culture is a biotechnological technique where small pieces of plant tissue, called explants, are grown on a nutrient medium under sterile conditions to produce new plants. The nutrient medium contains minerals, vitamins, growth hormones, and sugar to support growth. It is based on the principle of totipotency, the ability of a single plant cell to regenerate into a whole plant. Uses: rapid plant multiplication, conservation of rare/endangered plants, production of disease-free plants, genetic modification and crop improvement. Micropropagation. Micropropagation is a specific application of plant tissue culture used for rapid clonal propagation of plants in large numbers. It allows identical copies, clones, of a superior plant to be produced quickly. Commonly used for crops, ornamentals, and medicinal plants. Advantages: mass production in limited space, year-round cultivation, independent of season, uniform and disease-free plants. Techniques of plant tissue culture. There are several techniques, depending on the plant part used and the goal. Callus culture. Explants are placed on a medium with growth regulators. Rightward arrow form an undifferentiated mass of cells, callus. Cell suspension culture. Callus is transferred into a liquid medium and shaken to disperse cells. Useful for producing secondary metabolites, example alkaloids, flavonoids. Protoplast culture and fusion. Cell walls are removed enzymatically. Rightward arrow naked protoplasts obtained can be fused to create somatic hybrids. Embryo culture. Immature embryos are cultured to rescue hybrid plants, overcome seed abortion. Anther and pollen culture. Produces haploid plants from pollen grains, which can be doubled to produce homozygous diploids, important in plant breeding. Organ culture. Direct culture of plant organs like roots, shoots, or embryos. Process of plant tissue culture, steps. Selection of explant. Small part of plant, leaf, stem tip, node, root, etc., chosen. Sterilization. Explant and tools are sterilized, using ethanol, sodium hypochlorite, autoclaving. Inoculation. Explant is placed on sterile nutrient medium under aseptic conditions, usually in a laminar airflow cabinet. Callus induction. Explant develops callus on a medium containing auxins and cytokinins. Organogenesis/somatic embryogenesis. Callus differentiates into shoots and roots, or directly forms somatic embryos. Plantlet formation. Small plantlets develop in the medium. Hardening. Plantlets are transferred to soil or greenhouse for acclimatization before field transfer. Chemicals required for plant tissue culture. Plant tissue culture medium must provide nutrients, vitamins, hormones, sugar, and gelling agents for plant growth. The most widely used formulation is Murashige and Skug, MS, medium. 1 macronutrients, major salts, needed in large amounts. Provide nitrogen, phosphorus, potassium, calcium, magnesium, sulfur. Nitrogen sources. Ammonium nitrate, NH4NO3. Potassium nitrate, NO3. Phosphorus. Potassium dihydrogen phosphate, KH2PO4. Potassium. NO3, KH2PO4. Calcium. Calcium chloride, CaCl22H2O. Magnesium. Magnesium sulfate, MgSO47H2O. Sulfur. From MgSO4. 2 micronutrients, trace elements, required in small amounts. Essential for enzyme activity and metabolic processes. Boric acid, H3BO3, boron. Potassium iodide, Ki, iodine. Manganese sulfate, MnSO4H2O, manganese. Zinc sulfate, ZnSO47H2O, zinc. Sodium molybdate, Na2MoO42H2O, molybdenum. Copper sulfate, CuSO45H2O, copper. Cobalt chloride, CoCl26H2O, cobalt. Ferrous sulfate, FeSO47H2O, iron. Ferric EDTA, Na2EDTA2H2O plus FeSO4, iron chelate. 3 vitamins and organic supplements. Thiamine HCl, vitamin B1. Pyridoxine HCl, vitamin B6. Nicotinic acid, niacin. Glycine, amino acid. Inositol, growth factor. 4 carbon source, energy supply. Sucrose, 20 to 30 grams/liter, most common. Sometimes glucose or maltose used. 5 plant growth regulators, PGR/hormones. Balance of auxins and cytokinins determines growth pattern. Auxins, root induction, callus formation. 2,4-D, 2,4-dichlorophenoxyacetic acid. IAA, indole 3-acetic acid. NA, naphthalneacetic acid. EBOT, indole 3-butyric acid. Cytokinins, shoot induction, cell division. BAP/BA, 6-benzylaminopurine. Kinidin. Zetin. Gibberellins, GA3, stem elongation, breaking dormancy. Abscisic acid, ABA, stress response, embryo maturation. Ethylene releasers, rarely added, usually controlled naturally. 6 gelling agents, solidifying medium. Agar, most common, 0.8 to 1.0%. Agarose, high purity. Gilrite/Phytogyl, alternative gelling agents. 7 other additives, when required. Activated charcoal, absorbs toxic substances, darkens medium. Coconut water, banana pulp, casein hydrolysate, natural supplements in some protocols. Antibiotics, to prevent contamination or select transformed tissues. These chemicals are combined in different ratios depending on the plant species and purpose: callus induction, shoot regeneration, root formation, or secondary metabolite production. Explant culture. Explant culture is the in vitro culture of a small piece of plant tissue, called an explant, on a nutrient medium under sterile conditions to regenerate a whole plant. Explant equals any part of a plant, leaf, stem, root, shoot tip, node, anther, embryo, etc., taken for culture. Explants are cultured because of the principle of totipotency rightward arrow. Each plant cell has the genetic potential to develop into a complete plant. Steps of explant culture. Selection of explant. Chosen depending on the purpose, example shoot tip for virus-free plants, anther for haploids, leaves for callus. Surface sterilization. Explant is sterilized with chemicals, ethanol, sodium hypochlorite, mercuric chloride, to remove microbes. Inoculation on nutrient medium. Explant is placed on sterile nutrient medium, usually MS medium, inside a laminar airflow cabinet. Callus induction. On medium with auxins and cytokinins, the explant starts dividing and forms callus, undifferentiated mass of cells. Organogenesis/somatic embryogenesis. The callus differentiates into shoots and roots, organogenesis, or directly forms embryos, somatic embryogenesis. Plantlet formation. Shoots and roots combine to form a small plantlet in culture. Hardening and acclimatization. Plantlets are transferred to soil/greenhouse to adapt to natural conditions before field planting. Types of explant culture. Based on the explant used. Leaf culture. Stem or nodal culture. Shoot tip/apical meristem culture, virus-free plants. Root culture. Anther/pollen culture, haploid production. Embryo culture, rescue of hybrids. Applications of explant culture. Rapid clonal propagation, micropropagation. Production of disease-free plants, from meristems. Conservation of rare and endangered species. Production of secondary metabolites, alkaloids, flavonoids, etc. Genetic transformation in crop improvement. Study of morphogenesis and totipotency. Applications, products, advantages, and limitations of plant tissue culture. Applications of plant tissue culture. Micropropagation, clonal propagation. Rapid multiplication of identical, disease-free plants. Widely used in orchids, bananas, sugarcane, potatoes, strawberries, etc. Production of disease-free plants. Meristem culture helps eliminate viruses and pathogens. Conservation of rare and endangered plants. Tissue culture allows in vitro conservation, germplasm storage, cryopreservation. Plant breeding and crop improvement. Haploid plants from anther/pollen culture, rightward arrow doubled haploids equals pure lines. Somaclonal variation, rightward arrow new traits. Protoplast fusion, rightward arrow somatic hybrids, example potato times tomato. Secondary metabolite production. Plant cell cultures produce pharmaceuticals, alkaloids, flavonoids, etc. Example, shikanin, agmelicin, taxol. Genetic engineering support. Provides regenerable cells/tissues for agrobacterium-mediated transformation or gene gun delivery. Embryo rescue and overcoming barriers. Embryo culture saves hybrid embryos that would normally abort. Research tool. Study of plant physiology, morphogenesis, totipotency, stress responses. Products of plant tissue culture. Plant tissue culture is not just for research, it produces valuable products for agriculture, medicine, horticulture, and industry. One-whole plants. Micropropagated plants, large-scale production of clones, bananas, orchids, sugarcane, potatoes, strawberries. Disease-free plants, from meristem culture, potato, banana, citrus. Rare/endangered species, conserved and multiplied in vitro. 2 secondary metabolites. Bioactive compounds produced by plant cell cultures, used in pharmaceuticals, cosmetics, food additives. Examples. Taxol, paclitaxel, anti-cancer drug from Taxus, U-tree. Agmelicin, vincristine, vinblastine, anti-cancer/anti-hypertensive alkaloids. Shikanin, used in dyes and medicines. Capsaicin, from chili cultures. Vanillin, flavoring agent. 3 somatic hybrids and cybrids. Produced via protoplast fusion. Example, Tomato, potato times tomato. Useful in crop improvement, disease resistance, stress tolerance. 4 haploid and double haploid plants. From anther and pollen culture. Used in plant breeding. Rightward arrow, rapid development of pure homozygous lines. 5 artificial seeds, synthetic seeds. Somatic embryos encapsulated in protective coating. Easy storage, transport, and large-scale propagation. 6 genetically modified, GM, plants. Tissue culture provides regenerable cells for transformation. Products. BT cotton, insect resistant. Golden rice, vitamin A enriched. Herbicide resistant crops. 7 virus-free germplasm. Produced by meristem culture and stored for global exchange. 8 biopharmaceuticals, plantibodies and edible vaccines. Plants engineered in culture to produce antibodies, plantibodies. Edible vaccines, example against hepatitis B, cholera, rabies in banana, potato, tomato. Products of plant tissue culture in daily life. 1 food and agriculture products. Banana rightward arrow, disease-free, micropropagated plants, widely cultivated. Potato rightward arrow, tissue culture ensures virus-free seed potatoes. Sugarcane rightward arrow, rapid clonal propagation for high-yield varieties. Strawberry, pineapple, apple, citrus rightward arrow, high-quality, uniform fruits. Orchids and ornamentals rightward arrow, tissue culture used for commercial flower industry. 2 medicines and pharmaceuticals. Taxol, paclitaxel, rightward arrow, anti-cancer drug from taxa-cell cultures. Vincristine and vinblastine rightward arrow, anti-cancer drugs from Catharanthus roseus. Agmelicin and reserpine rightward arrow, anti-hypertensive drugs. Chicanin rightward arrow, used in wound healing, dyes, and cosmetics. Artemisinin rightward arrow, anti-malarial compound from Artemisia cultured cells. 3 flavors, colors and additives. Vanillin, flavoring for cakes, ice creams, chocolates. Capsaicin, spicy compound from chili cultures, used in food and pain relief creams. Chicanin, natural dye for cosmetics, foods. Menthol, flavor in toothpaste, chewing gum, candies. 4 household and commercial plants. Ornamental plants, orchids, ferns, palms produced by micropropagation rightward arrow, used in homes, offices, hotels. Timber and plantation crops, teak, eucalyptus, bamboo, sandalwood rightward arrow, raised through tissue culture for furniture, paper, and construction. 5 biopharmaceuticals and vaccines. Edible vaccines, in research/field trials, example hepatitis B vaccine in banana/potato. Plantibodies, plant-made antibodies used in diagnostics and therapies. Advantages of plant tissue culture. Rapid multiplication, thousands of plants can be produced from a single explant. Year-round production, independent of seasonal or environmental conditions. Uniform plants, all clones are genetically identical to the parent. Disease-free plants, meristem culture eliminates viruses and pathogens. Space efficiency, large number of plants grown in small lab space. Conservation, rare, endangered, or difficult to grow species preserved. Breeding aid, produces haploids, hybrids, and allows faster development of pure lines. Commercial value, used in horticulture, forestry, agriculture, and pharmaceuticals. Limitations/disadvantages of plant tissue culture. High cost, requires expensive equipment, chemicals, and sterile conditions. Technical expertise, needs skilled personnel for success. Contamination risk, microbes can quickly destroy cultures. Somoclonal variation, sometimes plants are not true clones, undesired variation. Hardening needed, plantlets must be acclimatized to outside conditions, often fragile. Not always genetically stable, long-term cultures may develop mutations. Species-specific responses, not all plants respond well to tissue culture protocols. Scalability issues, large-scale production still limited in some cases compared to field propagation. Thanks for watching.